Thank you. It's an absolute honor to be here with you this afternoon to talk about some of the currently enrolling clinical trials of immunotherapy for kidney cancer. So these are some of my disclosures, advisory boards of which I've participated in, and research support in terms of funding and sometimes drug from these two companies. So I think in no doubt, cancer immunotherapy has seen a major resurgence of interest in the last several years. How I think about cancer immunotherapy is really an indirect targeting of the tumor via revving up or re-engaging our immune system. And in 2013, Science noted cancer immunotherapy as the breakthrough of the year. And what you see here is sort of the T cells right here on the attack of the tumor. And so some of the rationale behind using immunotherapy is really that the tumor has developed mechanisms to evade our immune system, or in other cases really actually just looks too normal for our immune system to recognize it. And so we can give therapies that actually can re-educate our immune system to recognize these tumors as foreign. And you heard a lot about this morning, things like antibodies, vaccines, cytokines, chemicals that our own immune system can produce that actually can either stimulate or inhibit our immune system. And then here I've got, you saw some fun pictures this morning. Sometimes, for some reason, we always want to use a gun with the killer T cell here. But T cells really have great potential, not only to recognize antigens, which are really these abnormal proteins or mutations on the tumor cell, and then mount a specific immune response to this certain protein, and that actually, in order to kill the tumor. And that specificity also can result in less off-target toxicity. You also heard a little bit about memory cells this morning, and that's another great thing that T cells have the potential to do, which is to develop memory that can persist, and if tumors rise again, to kill them. So the brief outline of my talk today is really going to be about some of the T-cell targeted therapies for RCC. You heard a lot about PD-1 pathway blockade, and I'll put that into the specifics of renal cell carcinoma, talk about up-and-coming combinations, whether it be with our standard therapies like the VEGF inhibitors, CTLA-4 blockade like you heard about with ipilimumab in melanoma, and other agents with different mechanisms of action. And then touch on two vaccine approaches, that of IMA 901 and AGS 003. So just to refresh, you've seen different forms of this morning, but what are we doing with this whole PD1, PDL-1 interaction? So we have our killer T cell that we want to wake up and be activated, and you have your tumor cell. And as you heard today, tumor cells can either automatically or intrinsically express that protein PD-L1 on their cell surface, or sometimes they actually upregulate it in response to the interferon gamma that's produced by the killer T cell. And so this interaction, if the antibody isn't there, basically turns off the T cell. So all of these antibodies have been developed to either block the receptor PD-1 or the ligand. And when this happens, we get two signals that basically activate the T cell, increased killing, and that's what we think is going on with the effectiveness of these drugs. And so over the last several years, we now have what is actually a good thing, a laundry list of different PD-1 and PDL-1 blocking agents, mostly antibodies, but also one down here that's, right here, that's a fusion protein. And the ones that are farthest along, you'll hear me talk about today, nivolumab, which is a fully human PD-1 antibody, and MPDL 3280A, which is an antibody to PD-L1. You'll also hear about pembrolizumab. It's had a variety of names over the years, and it's also an antibody to PD-1. But all of these are in various stages of development, from first-in-human trials to now combinations with the more developed ones. So just a brief summary of the one that's the most advanced in renal cell, and that's nivolumab. In the phase 2, there was a phase 2 study of over, I think, about 160 patients that was presented earlier this summer. And basically showed that it overall had a very good safety and tolerability profile. In the patients that were treated, we had some promising efficacy in terms of upwards about a 20% objective responses, meaning significant shrinkage of tumors, durable responses, and overall survival in these somewhat heavily pretreated patients that had had two to four lines of therapy at close to 2 years. So the overall survival was especially promising. What you see here is actually the phase 1 study, but something that you're going to probably see a lot of today, these so-called spider plots. And line 0 is the patient's baseline tumor burden, and anything below that means some degree of tumor shrinkage. Anything above it means some increase. And you see the great majority of people had shrinkage of their tumors. This is that greater than 30%, which we really, really like to see. And the other really great and notable thing with this agent is the duration of responses. These are patients that are out 140 weeks since the start of treatment. And that's what's really notable with these drugs. So in order to get FDA approval, we need to do these big phase 3 randomized trials where we pit the new drug compared to an old standard. In this case, nivolumab was compared to everolimus, which is a second-line oral mTOR inhibitor. And so patients with clear cell RCC who had had 1 or 2 prior VEGF inhibitors were enrolled. Over 800 patients have already been accrued and were randomized to either the nivolumab every 2 weeks or everolimus. And the primary endpoint is going to be overall survival, and sort of the usual suspects for secondary endpoints of response and progression-free survival. Some interesting looks at whether that PD-L1 expression on the tumor actually correlates with response as well. So we all eagerly await the results of this trial in the coming year or two. But another reason, as I'm going to talk about, enrolling trials, is why should we take these agents further in development in RCC? And I want to just really highlight some of the distinct features. So those durability of responses, they can really be long-lasting, even when patients have stopped therapy, either because of tolerability or other reasons. We've sometimes even seen responses that started after the drug was stopped. And this also suggests this induction of a memory response specifically against the tumor. And we just simply do not see that with our traditional targeted therapies or chemotherapies. We also have these so-called unconventional or delayed immune-mediated responses. So one of the earlier sessions noted it, especially with ipilimumab, where early on, you might see actually an increase in the size of the tumor. And traditionally, we would have said, oh, God, no, this is not working. But as actually Rusty's experience showed in melanoma, when they removed one of his metastasis, it had showed all complete necrosis. And so this may be one mechanism by which, by imaging, it looks bigger, but it's really because of either necrosis from tumor cell death or actually an infiltration of all the immune cells makes it look a little bit bigger. And sometimes immunotherapy simply takes longer. It takes longer to rev up our T cells for them to proliferate and take effect than our other traditional drugs or chemotherapies that just go in and kill anything, okay? And then I think an important thing, as you'll see, and I'll talk about what monotherapy versus combination, is that tolerability of these drugs make it a good partner compound. And finally, while we're really excited about these drugs, we do have to remember that there are still a fair amount of patients who do not respond, at least by our traditional response criteria. And so that also kind of suggests we might need to use them in combination with other effective agents, or maybe, as I said, immunotherapy can take longer. We maybe can't just wait for that traditional response criteria or progression-free survival. We might need those overall survival endpoints. It may be a better marker of effect with these agents. And so, speaking of combinations, rarely is any cancer, be it RCC or others, cured by one drug alone. So I think combinations make a whole lot of sense. And the most rational thing would be to combine it with a drug that you already know works. And so we have now 7 targeted therapies in renal cell carcinoma that are FDA approved, many of which are these so-called VEGF inhibitors, whether they're oral drugs that are targeting the receptor or the IV antibody bevacizumab. And so these are established agents. They have different mechanisms of action than PD-1 blockade, so this makes sense. Also, they may actually be immunotherapies themselves, the VEGF inhibitors. They can actually create a more hospitable immune environment. And this has been shown in various studies that VEGF actually suppresses our dendritic cells. Those are the antigen-presenting cells that recognize the tumor, process it, and then present it to the T cell. The drugs like Sutent have also been shown to decrease cell populations that are immunosuppressive, those regulatory T cells, or Tregs you'll hear us say, and myeloid-derived suppressor cells. And finally, whenever we think about a combination, we have to say, is the degree or amount of increased effectiveness worth all the extra toxicity? So we'll think about that as we move forward. So our first hint of sort of the results of combining nivolumab with a VEGF inhibitor, sunitinib, came at ASCO this year, early this past June. And what we saw here was, just to refresh your memory, the phase 2 nivolumab monotherapy cohort in RCC. Here's, again, a waterfall plot. So 0 means the baseline. Anything above means increase. Anything below means decrease in the patient's tumor burden. And what we see here is, upwards of 40% of patients not having any response to the therapy. But when they combined it with nivolumab, you see a significant decrease in almost all of the patients. I think there was one patient that didn't have a decrease. And so this lack of primary tumor escape is especially promising when we add these two drugs together. However, again, what about the toxicity? And yes, they are more toxic together. I didn't show you the data, but another arm of that study combined nivolumab plus Pazopanib or Votrient, and that was too toxic. It didn't make it into the next level of testing. What we do see is an increase in toxicity or irritation of the liver to the kidney, skin toxicity. 70 to 80% of patients had high-grade toxicities, but they were generally manageable. And 36% of patients had to discontinue the combination. So this is going forward in further testing, and I think it'll answer the question of efficacy, and then we can weigh that with the toxicity. So another smaller study that has sort of resulted preliminarily is combining the MPDL drug, which is an antibody to the ligand, the PD-L1 ligand, plus bevacizumab, that IV antibody to VEGF. And here they were enrolling 20 patients. They presented data on 10 of them. Overall, they really found it was quite safe and tolerable, no unexpected toxicities, and they saw a hint of exciting efficacy. So here, that spider plot again, the different colors are actually them rating the degree of PD-L1 expression on the tumor. And what you see here is four of the 10, so 40% of patients had significant decreases in their tumor burden, more than 30%. Another 5 patients had sort of stable disease, and 1 patient had that was a non-responder. His tumor was a non-responder. And so this is interesting. We need to finish the 20 patients to make sure it's safe. And if it's safe, the plan is to go to a randomized trial in patients that have not had prior treatment for RCC. Patients will be randomized either to the PD-L1 antibody alone, the combination with bevacizumab, or the standard of care, sunitinib. And so this will be a good study to really tell us whether the combination or even the monotherapy has a greater progression-free survival than our standard drug, sunitinib. Going back to antibodies that target the receptor, pembrolizumab. This study also has a phase 1 component ongoing where they're looking at combining it with Pazopanib. Now remember, nivolumab couldn't easily be combined with Pazopanib, but maybe pembrolizumab is slightly different and will be more tolerable. In general, Pazopanib is a more tolerable drug than sunitinib or Sutent, but it can have more liver toxicity. And so this study, once we find out what is the most tolerable dose of Pazopanib, the standard dose of 800 or a little bit lower at 600, we'll combine it with pembrolizumab up here, MK3475 is the old name, versus Pazopanib alone or pembrolizumab alone, and with the primary endpoint of progression-free survival. So this isn't quite open yet, but will likely open soon. And finally, just to touch on another one, you'll see it's sort of almost becoming an alphabet soup of combining each of these different PD-1 antibodies and PD-L1 antibodies with the different VEGF inhibitors and figuring out which one is the best and which is the most tolerable. And so Exitinib is a second-line VEGF inhibitor that is actually a little bit more selective than sunitinib and Pazopanib, and so it may have less toxicities and be more tolerable. So it makes sense to actually to test this with pembrolizumab. The other reason it made sense was the data from the study that got its FDA approval in the second line, where patients who had had prior cytokines like interferon and high-dose IL-2, but not a prior VEGF inhibitor, actually had a median progression-free survival of 12 months compared to the comparator there of sorafenib of 6. And so this sounds like a good drug to try to see if we can increase its effectiveness in the first-line setting by adding pembrolizumab. So that's ongoing.